Anyway, over to Ross and Rum Jungle.
Hello again. Yes, so let's go through another case study to give an idea of how you can do some pragmatism in these systems and end up with something that can kind of work. So a little bit of background in this, we'll end up with some funky diagrams about how we've come up with revised site-specific water quality guideline values, which are we call locally derived water quality objectives, just because we did it slightly ahead of UNCAN's becoming ANZG. Rum Jungle is a rehabilitation project. We're trying to rehabilitate a mine that's already been rehabilitated, but it's a government legacy mine. So we already had inherently set it up under the PSER framework of pressure, stress, so physical, chemical measurements. I'm not pressing buttons, but it's moving. And I don't know whether I'm or the crocodile look more surprised in that picture, but we do actually measure a whole range of biota in the system, as well as measuring the physicochemistry and particularly toxicants. The main toxicant in this system really is copper, but there are a number of other co-occurring metals in particular of concern in a highly acidic system. Initially, we had set the community values and it included for all of the zones of the river that we divided up into, aquatic ecosystems and culture and spiritual values were equally important for all zones. Other community values came in and out depending on where you were in the system. We initially, on the basis of that and discussions around level of protections with the stakeholders, set water quality guideline values that were based on the national default guideline values. But they didn't work very well in this system. It has elevated concentrations. It's a legacy system, and that didn't work all that well for us. So even though we had effectively encompassed culture and spiritual values and aquatic ecosystem community values for each of these zones, and we'd had communication with the traditional owners around how we could use ecosystem protection values to protect culture and spiritual values, and they were comfortable with the levels of protection that we had set and with the level of ecosystem response to those levels of protection. We even showed them species sensitivity distributions so that they understood what groups of organisms tend to be sensitive and responding at those sorts of concentration levels, and how that affected their cultural and spiritual values of the ecosystem components. We still found that those numbers were not particularly useful in this system because it's naturally mineralized, because it's also a legacy mine, and because it had been impacted. What we did have was a gradient of impact because it's a legacy system and it's leaking acid drainage into the system that varies between years and over time within the wet season and dry season. It meant that we could have a look at what could that local ecosystem respond to. And so what we did was, knowing that we were measuring in each year of biological assessment, we were looking at all of the fishes in the system. We're looking at the macroinvertebrates in the system. We're looking at the periphyton in the system. So we had a multiple taxonomic group biodiversity database that we could compare with the monitoring data sets that are all for the physicochemical stresses as well. And we can compare the two. So really, with a little bit of statistical analysis, we're able to fit dose response curves of the measured ecosystem biodiversity richness values. There's a few more adjustments behind all of this. Probably won't go into that unless people want to question me about the relationship in specifically. But for example, we got very good relationships for copper, for nickel, for sulfate, and a few other parameters that are responding to the ecosystem's response to the concentrations of those particular parameters in the environment up to the time of taking the biological measurements. So these are based on in situ biodiversity measurements versus in situ measurements of water quality. And from those, we could then fit curves, and from those curves, we could derive numbers that are based on the levels of protection that we had applied to each zone in the river. Those numbers were then taken back out to the stakeholders, and in the stakeholder consultation, they were all approved. A much longer process with the stakeholder that always matters the most, in this case, both the territory and the federal environment departments. And we had that conversation over about a 7-year period, but we've eventually now managed to have these written into the rehabilitation approval process. Not quite how we'd intended it, but they are actually approved. All of this is available within the Rum Jungle Rehabilitation Plan project website. So it's all up there, and I'm happy to take any questions about how we use these in situ numbers to derive guideline values, if anybody wants to ask questions. Thank you.
Any other question? Let's just move on. We've got a little bit of time pressure because the fly's got to catch a plane. But what we're going to do is shuffle things around a little bit, but we've got to get through GVs for receptors, and then we're going to move weight of evidence for monitoring. That's all, and hopefully we'll get it all in. How much is in the receptors? GV for receptors, a lot. Guideline values for receptors. That is yours, my friend. And back to Chris. Thanks, mate.
Oh. So we've got our few streams of ecosystem receptors, and we've been talking about biological effects-based approaches from toxicity. So really, the toxicity line of evidence has already been covered. So we're getting guideline values from toxicity, and sometimes that's field effects approach as well. For bioaccumulation and biodiversity, the guidelines from 2000 and the current guidance is more about, meeting certain decision criteria and the effect size, the minimum departure away from your identified management goals. And in this case, because we're trying to maintain management goals, we're basically saying that we don't want departure from that particular condition, because that means that we're not meeting the management goals. But in order to be confident about that decision, we need sufficient data to ensure that we're not reaching an incorrect conclusion. And so we need to ensure that there's sufficient replication to that we're not going to incur a type 1 error, inferring an impact when there's none, but particularly inferring no impact when in fact there has been an impact. So, and those trade-offs are negotiated. So that particular advice has been entrenched since 2000 and carries over. So it's more about some of the other considerations when it comes to biological indicators in that space. And Ross and I have toed and fro on the seasonal sensitivity stuff very fruitfully. There's some information that can assist in interpreting biological data, some tolerance databases, for example. And Shelley's going to talk about some other aspects as well in recruitment timing. I'll just touch on this seasonal sensitivity stuff because it's something that Ross first introduced back in 2004 in an ACMER mining people handbook. So the concept is really, are there periods of the hydrograph that confer greater tolerance or conversely greater sensitivity to water quality to aquatic biota. And I guess the key question for me really is, are the guidelines sufficiently protective across the hydrograph? So, there's a couple of ways you can do this, and Ross and his colleagues in 2004 compiled all of their thinking across a number of water types and conceptualized their understanding of seasonal sensitivities. So that was, I think, Ross, you used a mix of good professional judgment and actual data. Whereas we actually took that a step further in the last few years up in Magilla Creek, and we actually did some acute toxicity testing of the type that Ben Kefford has done, rapid toxicity testing, grabbing samples from the stream bed and taking them back to the lab and exposing the aquatic insects to, in this case, magnesium. So the problem is with aquatic insects is that they're quite difficult to culture in laboratories, and they're typically not included in deriving guideline values and creating SSDs. So this is another approach that Ben Kefford came up with, and we've sort of continued that, but in quite a robust way, we think. So we've got sort of two comparisons here. One, the original conceptualization from Ross, and the one that we've done, which is quite empirically based for this particular system, Magilla Creek. So there's Ross's conceptualization. So that dashed line is a line of conceived tolerance. So the higher tolerance is, in this case, very early in the hydrograph, even probably before the system is even inundated. But then with inundation and exposure of those sort of hatched propagules, your system suddenly becomes much more sensitive. And that carries across the hydrograph while there's peaking flow, of course. And, later in the wet season, you can even get, a decrease in pH as a consequence of increase in organic tannins and so forth. So, pH drops a little bit, and your sensitivity can go up because some of your contaminants are mobilized by lower pH. But then the system dries out, and then you're left with a lot of adult, like aerial breathing organisms. Then a lot of those, I guess the crustaceans, microcrustaceans and so forth will diapause and go back into the sediments. So in terms of what Ross was looking at, it turns out that was a fairly broad suite of systems, but for the convenience of this particular conceptualization, I like to think of it as maybe as a system with more limited refugia, and particularly compared to what I'm about to show in terms of Magilla Creek, a bit more isolated from refugia in the non-wetted phase. So you could conceive this as something, very realistic for an arid, semi-arid, or small order stream. And I think even for the wet dry tropics, Ross, that was it the east branch of the Finnis that you had in mind? No, not in those days. That's correct. Yes. So I think that explains that enough. So then we'll look at the other one that we derived, and it looks quite different. So we start off with some quite sensitive organisms at first flow. So there's no opportunity there really. So in Ross's example, yeah, there might be that could be very quick, by the way, that, you know, there's a period in which the encysted or the propagules are actually hatching and coming into the surface waters. But here we've got something quite different, and that is that the system is very sensitive at the outset. So as material flows from upstream, you know, or at first flow, it's derived from upstream, and it moves its way downstream and infills the whole dry sand bed. So quite sensitive in the early phases. Like Ross's, you know, over time, the density, the abundance of organisms increases. And here we have, you know, a less sensitive phase here, and that drops off just slightly at the end of flow. But I guess the key difference is down here, that the recruitment is actually occurring along the front of water as it's moving downstream. We've got some permanent headwaters of flowing water, and the time that the water takes to get from those permanent waters, where all the rainfall comes from, that the stone country effectively, to the sites that we were sampling, could be up to a couple of weeks. But you've got the communities from those systems actually moving down the front, and there's a whole mix of you know, sensitive mayflies and caddisflies and early life stages. So yeah, so the other difference with Magilla Creek is basically we've got far more refugia than in these systems up here. So that's that. I'll just go a bit further. So how would we apply this? So yeah, I mean, it's useful to conceive these and then look at the background of your exposure, I guess. So if you've got hydrological data and pulse exposure data, you can indicate when your systems might be possibly more tolerant than other phases. For example, if there's a period that you've got adult aerial breathing organisms, you know, coinciding with a pulsing hydrograph, then the system is probably a bit more inherently tolerant. But a recessional flow, you know, as I mentioned at one of the very early talks, your dilution of wastewater to surface water and the groundwater discharge into the system might be reduced, so that you've got another period of sensitivity or recession. And as I mentioned for the first one, you can have periods of lower pH later in the hydrograph that could be sensitive to organisms. One other thing here was that Ross had, you know, originally was, you know, wondering about, well, where is your guideline value in here? Is it, you know, where across that particular horizontal axis might it be? And yeah, I guess there's the potential that you could have sort of bottlenecks here in particular, where you've got just newly hatched propagules in this system here that are very sensitive, and that's the dominant component of the fauna. And yeah, it's a worthy question of whether even the default guideline values are sufficiently protected. So it's something worth looking into. I guess the default guideline value in this, you know, we're talking here about microcrustacea and that sort of thing. And I guess at least with most of the default guideline values, we do incorporate microcrustaceans as whole life stages. But yeah, I think that's an area that needs to be looked at. I was just saying, Ross, when you're out, that, you know, this phase of the hydrograph, when you've got newly hatched propagules, You know, a valid question to ask is whether even the default, which we regard as quite conservative guideline value, is sufficiently protected. Yeah, and these are all based on macroinvertebrate data, and so there's a caveat on that, and you know, just because there's seasonal tolerance of one biotic group, that doesn't necessarily mean that it applies to others. I did mention in to and froing with Ross, sometimes in our systems we get pools of early filling, and they can be filled with tadpoles, for example. Ross pointed out as well that they can be quite tolerant to a number of stresses, but in other cases that's not always the situation. And so we've finished that bit. I'll just go to one slide that just in terms of some of the other tools that you can use in interpreting biological data. Organism sensitivities and tolerance sort of data that goes into biotic indices. And this is sort of a lot of the work that who's our author of our biotic indices? At a signal is Chessman, yes. Thank you, Shelley. It's an area that Bruce Chessman has been quite active in, looking at various biotic indices to predict sensitivity in particular systems, and he's recently come up with quite a very sensitive tool based on salinity and salinisation, particularly in these temporary systems, is an emerging stressor. So it's a good one to be up to speed on. Yeah, there's quite a number of suitable databases that are looked at salinity, and Bruce's most recent one is one particularly sensitive, and one that he's tested on independent data sets. So yeah, these are biotic indices that can be used to complement or to be used in the diagnostics of the biological information gathered. And I think Shelley, you might be next, Dave.
Sorry, everyone, my brain is now fried after a full week of conferencing. Okay, what we're largely focused on is, again, because we're working in that space of understanding what the risk may be if there has been a measured exceedance at a site within the receiving environment. For the most part, we can't rely on a lot of the published literature because, again, these streams are so ephemeral and temporary. It's that high level of dynamics, and a lot of the time we're actually suffering from the fact we've actually got multiple stresses coming into the system, and it's really trying to disentangle what may be the primary drivers of change in the system. So we've been working across a number of ideas to try and provide multiple lines of evidence, to provide a weight of evidence to say, yes, we may or may not be seeing an impact on the receiving environment. And for those of you who don't necessarily sit in the Queensland framework, the receiving environment is largely what the regulators use to characterise the systems that are downstream of a regulated activity. Those of you who have been at the conference this week will recognise that I have borrowed slides from Sarah's presentation on Wednesday afternoon. We collect macroinvertebrates using the Oz Rivers approach in these systems as part of our monitoring programs. A lot of the time, we've got to try and collate data from a number of systems to really try and understand, what a pseudo-reference condition might be, and to see whether we can assess whether there's actually going to be any deviations against different stresses or toxicants. So I'm just going to run through this really quickly. I'm only using sulfate as an example with this. Again, Chris has done all the hard yards with working through and developing all of this. I just get to stand up and talk about it. This is also coming out of the Queensland government guidelines, where there is an opportunity to use the 40th percentile of measured background conditions when you've got a highly modified environment as your starting point. And it just provides an alternative to the standard 20th, 80th percentile approach. So essentially what we have, hopefully you can see this, is up here is we have a fair amount of noise, and that's associated with our reference collections. So, and that's also at low concentrations, where it's also possible that there's actually going to be other parameters that may be influencing where our responses are sitting on that. Then we've got higher concentrations where we may or may not be seeing a response due to the parameter of concern. And in this case, as I said, we're looking at sulfate, but our indicator is actually the PET taxa. And so really what we're trying to do is do more with the data that we collect. So we do all the standard metrics around Oz Rivers. We'll do signal, we'll do PET taxa, but we're really trying to see if we can't tease out potential relationships between what we may have as stresses or toxicants actually passing through that system. This black line here, and so we've got the regression that's been put through this, and that's our 20th percentile of background there. So this black line here represents where our regression is actually passing through that threshold. As you can see, we've got sulfate values up to 8,000 there. In some cases, we still have some of the PET taxa present there, but by and large, we start seeing them disappear once we get above that threshold of around 200 milligrams per litre. Okay, so we're also going to look at, I've done it again, sorry. So we're also looking at the sensitivity. So we're doing exactly the same approach, but we're using the signal score of those and seeing how that falls out in terms of that threshold response. And as you can see up here, for our PET taxa, essentially, our trigger was around 200. For signal, it comes up to about 780 milligrams of sulfate. Why? That's a question we haven't been able to answer yet. So it comes down to whether some of the more sensitive signal species are actually slightly more tolerant to sulfate, or whether or not it might be another parameter that's actually driving a lot of this. And sulfate's an interesting one because it is both a toxicant and a stressor, but there can also be ameliorating effects embedded in that. So the challenge with this is that it can be difficult to tease out what the driver is. The advantage with this is that we're seeing what the effects are of sulfate in amongst all the other water quality parameters within these measured samples where we collected these taxa. And lastly is we're looking at the diversity. And again, we've got a mix. So in this case, PET taxa are much more sensitive to sulfate than our signal, and our richness is, you know, reasonably similar, but diversity has a different level of a greater level of tolerance. All it's showing is that we need to consider all these aspects in it, because if we took another parameter, it may be signal would be a more sensitive measure of that particular toxicant. So it's, if we don't collapse it all and look at all those, we may be missing what the drivers are around the system responses. This one here is something that we're just starting to try and do. We've been really fortunate. We've got a taxonomist that's come on board with us, and she's giving us that opportunity that when we collect our macroinvertebrate samples, she can just take it down to lower taxonomic levels. She can tease out the different instars, different stages, and it gives us an opportunity again to do more with the data that we're actually collecting. So this here is completely hypothetical. We're at the point where we're trying to collect that data and see whether we can actually detect these things. With all of these, there's a whole raft of caveats embedded into it. I'm not going to go into them, or we'll spend all afternoon on the caveats and not actually talking about what's happening. Essentially, if we get a rainfall event, and these rainfall events tend to be very, very sudden events, very short term, 12, 24 hours. If you're really lucky, we might have the rainfall event go out to 6 or 7 days, but that's pretty atypical for a lot of the sites that we're looking at. We're more likely to see a 6, maybe 12-hour event. Then you'll get the flow coming into the system. And the key thing around this is whether or not the flow that goes into that system is a flow event that reaches a point and moves through it, or if it's a highly erosive flow. And if it's a highly erosive flow, a lot of our beds are very sandy, mobile beds. And our biggest concerns are in those circumstances, what we're getting is an erosive flow, and it's actually dislodging anything that might be estivating, any cysts, any spores, any of the propagules, and it may be migrating those downstream so that they're not actually present. On top of that, we're close to the headwaters, so we likely may not have much opportunity for recruitment above where we're actually looking at these sites. As well, with the macroinvertebrates, we've got a significant proportion of external recruitment in there. So you get a lot of terrestrial adults that have macroinvertebrates that have larval aquatic stages. So the eggs get laid. So you need to have that time between flow commencing, recruitment of eggs, whether it's internal or external, and then looking at progression through those larval stages. So one of the things we're really keen to look at is where we've got in-stream recruitment, does that precede external recruitment in terms of hatching and development, or do they go hand in hand, and how much does erosive flows affect that? And in doing so, we also have at some locations a groundwater expression point that comes in after cessation of the rainfall event, and then all of a sudden you're getting a different water quality coming through in the system as you get that surface expression of the groundwater into the creek system. So is that creating its own clock reset? So it's really an exploratory process to see whether we can't tease out some of those aspects in looking at our data. And as I said, macroinvertebrate collections and sampling is a lot of time, effort, and money. And the more we can do with the data that we collect, the better off we are. And this also is very tightly tied back to a lot of the stuff that Chris was talking about in those previous slides, where, where are the sensitive phases in this? At that recruitment level, do we see, an initial recruitment break and then a secondary recruitment? If we've got erosive flows, that in-stream recruitment, does that actually have to migrate upstream? If we've actually dislodged it all? These are all questions we just don't have the answers to at the moment. This one's actually moving a little bit away from surface water and so on. Again, it's something that we're exploring around shallow groundwater. And the reason we're using that is that shallow groundwater is the groundwater system that is most likely to express into our surface waters after rainfall events. Now, groundwater, yes, it can be modelled. Bores will give us water quality. We can do stargofauna. I know there's been work being done in the eDNA space in that. But one of the things that we're looking at is, can we detect change in the shallow groundwater system using groundwater dependent vegetation? Now, the example I'm using here, these are terrestrial species. We are also doing some similar stuff with riparian species, but I'm not showing that one here. Sorry, my voice is, it's your fault, Ross. My voice is going to. Okay. And the example I'm using here is at a location where they do have a mineralized groundwater influence from a closed mine site. So we trawled through the literature and had a look to see if we could define what we might consider as groundwater dependent vegetation. There's a paucity of information out there in that space, but we did identify 2 species of trees that did fall into that category. Why did we choose trees over something else? Number one, they're still there at the end of the dry season. Okay, most of our herbs and forbs and grasses by that stage have just pretty much gone, and you've got little stumps left. At least this way, we've got something that we can actually sample and assess. These 2 species tap into groundwater around the 5 to 8 meters. So for this site, they were ideal. And what we did is we've got a number of separate sites that we consider either exposed to the mineralized groundwater or to background groundwater conditions. And we've got marked trees within each of these quadrats that we consider either control or impact. And as I said, we've got 2 species. Species A, we just don't have the same abundance of them. Species B, they're everywhere. We've been doing this for 5 or 6 years now, so we are collating a body of evidence. The interesting thing around this one is what we have found, and this data is from late 2020. And we're pretty much in a fairly significant period of water stress right throughout the region. And one of the really obvious things were a lot of species B in particular were doing a massive amount of leaf drop and looking horrendously water stressed, but we weren't sure whether it was water stress or it was a combination of water stress plus contaminant uptake. Why have I shown you silicon on this? Okay, so this is the historical data that we just claimed, and that was the current year data. And whilst you can see there's a little bit of variability in that, for the most part, there was quite elevated silicon. Going back through the literature, silicon expression is an indicator of water stress. So in that case, yes, what we were seeing is that we're seeing some fairly strong water stress indicators across this. I'm also just using copper. We do all the major nutrients. So we do carbon, nitrogen, oxygen. We try and get best bang for our buck when we do these samplings to maximize our data set. I'm just using copper as the example here. Again, what you can see, there's a lot of variance. But what we have got, and we haven't been able to tease it out yet, is that all of a sudden there was a bit of a spike in copper there. Now, we don't know whether that may have been a correlationship with that water stress. But what it is showing, certainly for this species, there is some indication that certainly for copper, we may actually be able to discriminate a copper signature if it's present in the groundwater. Biggest issue I've got is the bores aren't really closely related to my site. So there is a level of inference around these. Again, it's a bit of a proof of concept that we can still monitor shallow groundwater using biological indicators. And that's it. I think that's the end of my talking for today and this week.
Chris has a question. You can. Okay.
Just to add a bit of context to our invert data, one of the things is most of the waters we sample are pools. I'd say 70% of the time are pools less than 1 meter maximum dimension. So they're tiny little pools. So we can regularly get water samples. We can much more infrequently get invert samples. So one of the things we want to try and do is use our more infrequent invert data to provide some context to water only samples. So some of the regression analysis Shelley showed there is an attempt to move down that pathway. So we can turn water samples into biological effect samples.
Any other questions? Well, questions for Chris too. I realize he snuck off without being questioned. So if you've got something for him. We did cut into our timing a bit with that session. We thought we would. There was a lot of content. Sorry, Chris. You could. Yeah, go on.
No, it's just to Shelley. Have you thought of nursery trials and looking at seedlings in terms of uptake and stuff? But so was the purpose of that just to see that there was contaminant uptake?
We know there is a plume there. What we're trying to do is see whether we can find indicators that are going to detect migration of that plume, rather than just relying on bores and modelling. Yeah, modelling has its place, but we need to still be able to do some validation around this. So.
We've got a bit of time before afternoon tea, so we might move on to monitoring. I think I'll be able to squeeze my bit in, but we'll see if we can do it all. So the idea is, once we get all this to you, we will do some consolidation of thoughts and a bit of a, what are your thoughts on the gaps and ways forward and things like that. So we're really keen to get to that, but let's get through the monitoring.